we know about the molecule inside the cell cycle, inside the cell that can control the cell cycle. Um, okay, so as you know, the eukaryotic cell cycle is divided into 4 phases: Z1, the S, Z2, and the M phase. And the M phase consists of the mitotic and mitotic division and the nuclear division and the cytokinesis. So in each phase, the cell need to satisfy the condition so that they can move on to the next phase. And the status of the status that control the or gives the decision to the cell to continue or stop becomes a checkpoint. And in the Z1, this checkpoint is the most powerful checkpoint. They can call this a stop or the restriction point or the R point. So all the molecules that involving to the cell decision in the progress of the cell cycle or the stop of the cell cycle belong to the cyclin and CDK complexes. And there are many types of the cyclin CDK complexes. And each of the complex is responsible for the different stages in the cell cycle. So here is one in this figure you see some main cell cycle checkpoint during the from the Z1 phase to the M phase. In Z1 phase, the checkpoint mostly to give the answer of is environment favorable. That means during the Z1 phase, the environment factor is the main cause of the checkpoint on or off. And for the Z2, the checkpoint normally turn on if the cell has some mistake in the DNA replication, or if the cell find out some DNA damage, and they need to do the reparation. So the two checkpoints will be turned on. And during the end phase, the checkpoint normally will be turned on for the to be sure that all the chromosome properly attached to the mitotic spindle. So if there is some mistaken attachment, the checkpoint is turned on. And if all the chromosome is properly attached to the mitotic spindle, the checkpoint is turned off. And then the cell can move on from the metaphase to anaphase and finish the M phase to start to come in the new Z1 phase of the daughter cell. So the cell cycle control system ensures that key processes in the cell cycle occurs in the proper sequence and proper times. It's like right time and right place. So how the scientists figure out the cell cycle control? So as I say, that in the 19th century and 20th century, so the scientists observed the cell divide and stop divide, and they didn't know exactly what happened and which factor can induce the cell to come to the cell division and which factor can inhibit the cell division. So the many scientist group try to work on this um, to answer this question: which factor can induce the cell to divide? So here is one of the research group from Japan. This is a research group of Professor Kazuo Matsu's. Um, his group performed the experiment on the egg of the Xenopus. So Xenopus here belong to the frog. Okay, so Xenopus egg is, in fact, it is one very uh, very popular model to research about the early biological development. And it is one of the animal, the fertilized eggs of xenopus is suitable for the biochemical studies of the cell cycle because they are exceptionally large and divide rapidly. Here you see an egg from frog xenopus is over 1 millimeter in the diameter. So it's quite large enough for us to observe by our naked eyes. We don't need the support from the microscope to observe this egg or this cell. And after fertilization, it divides rapidly to partition the egg into many smaller cells. And this rapid cell cycle consists mainly of repeated S and M phases with very short or no Z1 or Z2 phases between them. As last week, I already showed you one video clip represent about the cell division as the early development. So you can see the cell cycle is very short and they must repeat the S and M phase without the G1 and Z2 phase. Because during this time there is no gene transcription and all of the mRNAs and most of the proteins required for this early stage of the embryonic development are already packed in the very last eggs. So that is why you see the egg is quite big and because they already store the molecules, the nutrient, and the other necessary molecule for the early development of the embryo. So, the Thai scientists think that the biological activity um, of the extraction from the dividing cells can be used to test if they can induce the other cell divide. So that is the thinking, the idea of the Keijo Matsui group, research group. When he observed the diving cell and the non-diving cell, so they think that in the diving cell, they must contain some factor that induce the cell come to the M phase. That means the cell division happen. So they take the cytoplasms of the M phase cells and inject this cytoplasm extraction into the interphase cell. Here is the outside of the non-dividing outside. And after the injection, they see the appearance of the spindle, mitotic spindle. So as you know at the last week, the mitotic spindle is one marker of the M phase cells. So that means the interphase cell is now stay in the M phase after the injection. And if he takes the injection from the interphase cell and inject it into the interphase cells, so there are nothing changed. That means the cell will not enter the M phase. So repeat again and again. They think that there must be some specific factor in the M phase cell cytoplasm that can control the cell to come into the M phase. And after that, they try to detect this factor and see. It turned out the factor have the activity of enzyme, of the kinase enzyme, and they call this factor is MPF. That means the maturation promoting factor because they work on the outside, so they call this is maturation promoting factors. And the year they find out this is in 1980s of the last century. And the activity of this kinase is changed during the cell cycle. The activity can increase very quick when the cell enters the M phase, and then in the late M phase, they decrease also fast, and then they increase again when the cell enters the next M phase. So the activity of the MPF changes during the cell cycle, and the activity increase when the cell enters to the corresponding phase in the cell cycle. One thing here is that when the other scientists try to purify the MPF, that means the kinase weak responsible for the cell come to the M phase. But when they try to purify them, they found that it always contains the other protein that requires for the kinase activity. And this protein is cyclin. And the scientific group who figure out the cyclin is a Tim Han group. This professor, he worked on the clams. Do you know clams? Clams console. Okay, so he used another model to do the research on the cell cycle control molecule, and Tim Han, by fixing in the clam, he identifies the M cyclin. So this protein have the concentration rise and fall gradually during the interphase. That means the cyclin concentration, they increase their concentration um, during the cell come from the interface to M phase. After that, they find out the concentration. In fact, the concentration of the M cyclin in fact start to increase from at the end of the S phase during the Z2 phase and get the peak on the in the M phase, and then they rapidly decrease at the latch of the M phase. And this protein repeats this performance in each cell cycle. So it's similar like the activity of the MPF, but the difference here is one changes the concentration and the other changes the activity. So that is the two things different between the cyclin and the CDK. But they always go together whenever the CDK have the activity. The his CDK here is MPF. Okay, the MPF has activity. So, in fact, the MPF that was discovered by Kazuo is a complexes of the kinase, the kinase and the cyclin. So the kinase is a catalytic subunit of the complexes, and the M cyclin is a partner of this. So after the MCDK, that means the MPF is identified. Another type of cyclin and CDK was isolated, whose concentration or activity, respectively, rose and fell at other stages during the cell cycle. So the other research group, like Hartwell or Sir Paul M. Nurse, they together figure out the other cyclin CDK complexes of the cell cycle, and together they got the Nobel Prize in 2001 for their discovery of the key regulator of the cell cycle. So different CDK and cyclin complexes will associate to different will associate with different cell cycle phases. Okay, so now we see there are two main key classes of the regulatory molecules that determine the cell cycle progressions. One belongs to the cyclin without the enzyme activity, and the other is a cyclin-dependent kinases. They call the CDK. And the when you read the name of the CDK cyclin-dependent kinases, you already understand that this kinase they have activity depend on the presence of the cyclin. So that is why they call the cyclin dependent kinases. And there are four classes of the cyclin, each defined by the states of the cell cycle at which they bind CDKs and function. And all eukaryotic cell requires three of these classes. And the other is for the I think all of yeah requires us for here's a mistake of these classes of the cyclin and CDK. So this is a list of the cyclin CDK complex and the corresponding phases that they control. So the CDK1 being uh, associated associated with cyclin A or cyclin B, and together they can control the cell during the mitosis. So they call them mitotic CDKs. CDK1 here is, in fact, it is MPF, or the other name is CDC2. The CDK2 uh, bind to the cyclin E and cyclin A, and together this complex will control the cell entry into the cell cycle S phase. And that is why they call this is Z1 S phase CDKs or S phase CDKs. The CDK4 will bind to the cyclin D and control the cell activity in G1 phase. And CDK6 also come to go with the cyclin D, and together with CDK4 cyclin D, they can control the cell in the G1 phase. So that is the four classes of the cyclin and CDK, and together they can control the cell in all the cell cycle. So why the CDK can depend on the activity of CDK depend on the binding of the cyclin? Because the binding of the cyclin to CDK will regulate the subunit, the CDK for the activity. So you see, I I don't know if you remember that one kinase, the activity of the kinase is transferring one phosphate group from ATP to their substrate to their corresponding substrate. So they must bind to the ATP and have the activity of the ATPase. That means to do the ATP hydrolysis to release one phosphate group and then transfer this phosphate group to their substrate. So they must bind to the ATP and also they must bind to their substrate. But normally, when there is no cyclin, the binding site of the ATP is covered by a loop. They call the T loop here, the T loop. So this T loop will cover the ATP binding of the CDK. So whenever there is cyclin, the partner, the suitable partner, so this cyclin bind to the CDK, and the binding of the cyclin to the CDK will change the conformation of the T loop. And now you see the T loop from this conformation to another conformation. And with the new conformation, they expose the ATP binding on the CDK. So that is the why, or that is the target of the binding between the cyclin and the CDK to control the activity of the CDK molecule. But it's not enough because only binding with cyclin is not fully active the CDK. So they need another way, another process like phosphorylation and dephosphorylation to have the full active of the CDK, which I've been telling you in the next slide. But for here, I would like to show that um, the cyclin is very necessary for the activity of the kinase of the CDK. And without the cyclin, the CDK have no activity. In the image below, you see this is one example of the function of the cyclin to the CDK. So the scientists they want to check the function of the cyclin to the CDK complex activity. So they make the ubiquitin relation of the cyclin. And inside the cell, it is one pathway that whenever the protein is ubiquitinated, so this protein will be degraded by the proteasome. So the scientists apply this pathway to the CDK complex. They ubiquitinated, they ubiquitinate the cyclin, and after that, the cyclin is destructed inside in the complex. So without the cyclin, the CDK have no activity. So this is a way they prove the function of the CDK in the of the cyclin in the CDK activity. And as I said, binding with a cyclin is not enough for the fully active of the CDK. So the activity of CDK are also regulated by both phosphorylation and dephosphorylation. Um, so the first state of the activating of CDK is the binding of CDK with a cyclin. After binding after binding with cyclin, the CDK will be continued to phosphorylated. But in this process, in this test, the phosphorylation happen in both the inhibitory phosphate group and the activating phosphate group on the CDK. That means in some amino acid site on the CDK, if this site is phosphorylated, it will inhibit the activity of the protein. And in the other amino acid site, in the if the other side is phosphorylated, they can activate the activity of the CDK. And if both sides are if both sides are phosphorylated, they can inhibit the activity. That means at this state, the CDK have no activity. And then they will move to the third stage, the dephosphorylation. So during this stage, the inhibitory phosphate will be removed by the phosphatase. And then after this step, the CDK is fully activated. So the normal process to activate of the CDK is they consist three stages: the cyclin binding, the phosphorylation, and the phosphorylation. Here is one example of this step for the CDK2 and cyclin B, which together control the M phase. So the scientists found that this CDK2 they need to bind the cyclin B first, and then they will be phosphorylated in on inhibitory and activating phosphate group. For example, the T14, Y15, T160. So the T here, that means the um, tyrosine, and Y is threonine, and T is tyrosines. So the tyrosine is amino acids at the site 14, the threonine at the site 15, and the tyrosine at the site 160. So these phosphates, these sites will be phosphorylated during the stage 2, and the T14 and Y15 they stay in the N terminal, and the T160 stay in the C terminal of the CDK. both the T14 and Y15 stay in the ATP binding pocket of the CDK, and whenever this site is phosphorylated, they can interfere with the positioning of the ATP in the pocket. So that is why they call it is inhibitory site. Okay, because whenever this site is phosphorylated, they will inhibit the ATP binding to this pocket of the CDK. But at the state 2, this site is both they are phosphorylated together with the T160. And after this step, the T14 and Y15 will be dephosphorylated. And after that, the CDK becomes fully activated. Okay, so also on the activation, during the activation of the CDK, there is another process called the positive feedback. The positive feedback here. So what is the positive feedback? The the positive feedback happen when the product of the reaction, in this case, is a CDK, the activating CDK. So whenever the CDK is activated, they can induce the process that activate them. So they call the positive feedback. How the CDK can do that? So if you see in this step, the third step or the third stage, when the CDK is dephosphorylated by the phosphatase, for example, the CDC25. So this phosphatase CDC25, they have to be activated by the phosphorylation, and the phosphorylation of the CDC25 is regulated by the CDK, by the activity of the CDK. So where after the CDK is fully active, this CDK they can induce the phosphorylation of the other CDC25. So the more of the active CDC25, the more of the CDK is activated through the dephosphorylation. So they make a loop, and they call the positive feedback because the product of the reaction induce the speed of the reaction. Okay, so that is why you see when the cell enter to the M phase, the activity of the PMPF or CDK1 increase very quick, very fast in very short time. That's because of the positive feedback phenomenon here. The activate CDK turn back to induce the speed of the reaction of the activating reaction for this molecule. So beside the activation of the CDK, the other control of the CDK is the negative control. That means the activity of the CDK can be inhibited by the inhibitor molecule in the cell. And there are two classes of the CDK inhibitor inside the cell. The inhibitor, the first one is the INK class. INK for class, that means the I and K, they call ink, like it's ink to write, right? So ink for class consists of the four member, the for A, for B, for C, and for D. So this ink class, they can bind to the CDK46 and distort the binding site of the CDK6, reducing its affinity to the D-type cyclin. So in the image on the right, they illustrate the binding of the CDK6 and their partner cyclin D can be interfered by the appearance of the P16INK4A, the CDK inhibitor. So if there is the INK4A here, so this protein they can interfere the binding site of CDK6 with the cyclin. So CDK cannot bind totally to the cyclin and the activity is inhibited. At the same time, when the ink binds to the CDK, they also distort the ATP binding site and thereby compromise the catalytic activity of the CDK. So similar, the second class they call the KEEP. The KEEP, there are two type KEEP, the KEEP2 and KEEP1. And a SIP1 also belong to the KEEP family. And this one, they can inhibit the activity of the CDK2 and CDK1. So this image is illustrated about the P57, P27. That means keep one. They bind, whenever they bind to CDK2, they also interfere the binding of the cyclin A to the CDK2 and also distort the ATP binding of this enzyme. So by this way, they inhibit completely the activity of the CDK2 during the C1 and the S phase. So this is another control of the CDK on the cell cycle. Uh, okay, we have a short break. Sorry, I forget the time. We have a short break and come back at 10:10.